Hey everybody, welcome to Biology Part B. We're going to start tonight with a little bit on the second half of the year. We're going to start in the unit on genetics and heredity, and we're going to go over the project on building DNA. Pretty easy project, pretty straightforward, but I just wanted to go over a little bit of the expectations for it and just talk a little bit about how it relates to the actual lesson that you did prior to it. So let's take first and talk a little bit about the objectives that the lesson is seeking out and then how that relates to the project. So hopefully after you've gone through the lesson, you'll be able to understand the differences between DNA and RNA because there are some very big differences. And then you'll get a better understanding of how DNA replicates, and then once you have an understanding of how DNA replicates, you'll be able to then explain how genetic mutations occur and really what the results of many of those mutations are. So let's take a few minutes and talk a little bit about some of the content of the lesson before we dive into the project here. Okay. Um, Going back to the first semester, we talked about the cell, the structure of the cell, and the organelles within that cell. Okay, and the most important being the nucleus, which is really the control center, the point where all cellular activities are really directed. Okay. And within that nucleus, the instructions for reproduction, for heredity, and cell functions are all right there in one central location. So you could think of it in an analogy form as the CPU, the central processing unit of a computer. Okay, and the DNA, the deoxyribonucleic acid, is really the software that contains all of the orders or the operations that occur within the program. Okay. Um, when you think about how genetics relates to that, genetics itself explains why all the traits in succeeding generations are inherited, from parent to child going down. Okay, um, but really, what your lesson focuses on were the main differences in terms of DNA and RNA. So um, remember that DNA really contains the highest concentration of data stored within any medium, if you will, known to humankind. So when you relate it to a computer, for example, a hard drive, um, those estimates vary, but the content that DNA has really between about 500 to 30,000 print books or volumes of information stored in it. So um, that is really insane when you think about it in terms of just sheer quantity of information. Okay, and with that means that the DNA is really the most important biological molecule found in living things. Okay, the coded information that it contains really determines the sequence of proteins, which really are your outward expressions, what you look like essentially as an individual. Okay, um, so with cells, the important thing that you really need to take into consideration is what's going on down at the molecular level in terms of what's going on with the DNA. Okay, so when you look at the structure of DNA, obviously it's helical in nature. Okay, it's basically like a spiral staircase. All right, and that twisted ladder, okay, has sides. Okay, or rails, if you will, that are made up of repeating units of the sugar deoxyribose and phosphates, high energy phosphate groups. Okay. And then the inside of that ladder are the rungs, which are the nitrogenous bases. And those are really the blueprint for life, if you will. Okay. Those are really what determine, okay, the information that will eventually become proteins. Okay. Now, when you compare DNA to RNA, okay, RNA is a little different. RNA really is it's single stranded, okay, and RNA is really what is made, it's a template made from your DNA that actually then is used to make the proteins that are the expressions that you will think of when you look in a mirror, essentially. Okay, but in order for that to occur, okay, you really need to go back to how DNA is replicated. Okay, and the replication process really occurs um, during the synthesis phase of the cell cycle, or the S phase. So for those of you who remember, back in part A, we talked about the cell cycle: gap 1 synthesis, gap 2, and then mitosis. Okay, when we're talking about the S phase of the cell cycle, we're talking about DNA replication. Okay, and replication occurs whereby those chains, or those rails on the twisted ladder, rip apart, open up like a zipper almost. Okay, and the bases are copied, thus forming two new strands from the templates. Okay, so when we refer to DNA that is being synthesized through replication, we refer to that as semi-conservative replication. Okay, because an original strand always serves as the template for the new strand. Okay, so with that, let's get in there and talk a little bit about the project now. Okay, so the project itself is entitled Building DNA, and let's go over to that project right now. Okay, so the project states that the structure of DNA resembles a twisted ladder called a double helix. And for this project, you're going to build a DNA molecule by placing its basic parts together like pieces of a puzzle. So back in the lesson, it talked about what they call Chargaff's rule. And that is the rule of base pairing that exists in DNA, whereby the base adenine pairs with the base thymine and the base cytosine pairs with the base guanine. So what you're going to do for this project, you're going to click the project, okay, and you're going to choose the molecules that make up the sides of the DNA ladder, and you're going to check it, and it's going to ask you a whole bunch of other questions up until the point where you're actually going to do base pairing. Okay. From there, once you're done, you're going to answer these two questions about the project. Okay, specifically, what kind of pattern do you notice with the structure of DNA? Okay, and if you get stuck, okay, go back to your notes on the lesson where it talks about DNA structure. And that paragraph, I want you to paraphrase that for me in a real brief paragraph. Tell me what you see in terms of what the rails and the rungs of the ladder represent. That is what this question is asking. And then the second question is briefly describe the DNA transcription process, which involves the transfer of DNA, the genetic code, onto a messenger, an mRNA molecule. So again, going back in the notes, all right, you're going to look at DNA replication. You're going to look specifically at transcription all the way up to and including translation. And you're going to there's 3 paragraphs. I want you to summarize those 3 paragraphs in about 3 to 4 sentences, explaining to me how DNA replicates itself, and then from that creates a template of messenger RNA all the way up through then a fully functional protein. All right, so those are the 2 things that this project is looking for from you. Do the video, or do the actual simulation, and then answer the 2 questions. And if you get stuck along the way, certainly by all means, come into the virtual office. Or I'm also going to post another video that goes over structure and function of DNA a little bit better than what the lesson does. So I would encourage you to watch that video as well. If you're still having problems, certainly seek me out. Be more than happy to help you. And we'll be back here next week going over a whole nother project. So have a great week, and we'll see you next week.